Good day, viewers, and welcome to your best learning hub for the sciences, Jalo Science Academy. I am Mohammed Jalo, and in this tutorial, I'll be taking you through your biology. And we're going to start off with digestion. So look at digestion in man. Now, when we talk of digestion, we are talking of the breakdown of food substances, okay, from the complex into simpler substances. And when we talk of digestion, digestion involves two processes. What are these two processes? Digestion involves a physical and a chemical process. And what is this physical process? Of course, this physical process here has to do with the physical breakdown of food substances, meaning the ingested food. When ingested into the mouth, it has to be chewed in the mouth. So one of the physical processes is mastication. Okay, the food has to be masticated so that the surface area of the food is going to be increased in order for enzymes to penetrate the molecules and then break them down into simpler substances. So the physical process here involves mastication in the mouth. And two, it involves the churning of the food, churning, which takes place in the stomach, churning of the food in the stomach. These are physical processes. So we refer to it as the hydrolysis. That is the hydrolysis. So we call it the breakdown of the food substances. This is for the physical process. Now, the chemical process here involves the action of enzymes. It involves action of enzymes. And enzymes, we know, are a biological catalyst, okay, that will help speed up a particular reaction. So this involves the chemical process. In the mouth, you have enzymes that are produced, which helps to convert the food from one state to another. And then inside the walls of the stomach, you also have enzymes that are produced, which helps to convert the food from one state to another. So these are the two processes that are involved in digestion, both the physical and the chemical process. The physical dealing with the hydrolysis of the food, and the chemical dealing with the action of enzymes. Now, we want to look at really where digestion starts. And when the food is ingested, so what do we mean by ingestion? Ingestion simply means the intake of food, intake of food into the mouth. So the solid food substance that is taken into the mouth, we refer to that as ingestion, meaning you are ingesting the food into the mouth. That's what we mean by ingestion. Now, when the food is being ingested, the food is going to be chewed in the mouth. So we look at digestion in the mouth. So what happens to the food inside the mouth? Now, inside the mouth, one of the physical process which we talked on earlier, mastication takes place. Now, as the food is being masticated in the mouth, what happens? Inside the mouth, you have glands. And these glands, we call them salivary glands. These salivary glands are of three. You have the submandibular, you have the sublingual, and you have the parotid. Now, these three here are found beneath the tongue. These two here are found beneath the tongue. And then this one here is found on top. Okay? on top, that is where it is positioned, and the two are positioned downward here. So when the food is being chewed by the teeth, these glands are going to secrete saliva, and the saliva, the secret, contains an enzyme. We call it amylase. Okay, it contains an enzyme called amylase, or we call it ptyalin. Amylase here is going to help in the conversion of the food. Amylase acts on starch. Starch, we mean they are carbs, the carbohydrates. It acts on carbohydrates, and it is very active on cooked starch. It is very active on cooked starch. Amylase here is going to convert this starch, which is a polysaccharide, it is going to convert it into another type of sugar, in that saccharide, we call it maltose. So amylase converts the starch into the maltose. And now when this one is being produced, inside the mouth as well, you have mucus that is produced. Apart from these salivary glands, you have mucus that is produced. And this mucus which is produced helps in the process of swallowing. It makes the food slippery. The food becomes slippery. Okay, so this slippery food is now going to be pushed down into the esophagus, and that is done with the help of the tongue, because the tongue is going to roll the food that has been chewed. It rolls it into a ball like this, and this one we call it the bolus. It rolls it into a ball called the bolus, and this bolus is going to be propelled down into the esophagus or the gullet. So let's look at how that happens. Now, when the food is being chewed into the mouth, it's going to be pushed in here. But before the food is pushed into the gullet lies behind the trachea. You have the trachea in front, that is the windpipe. So the windpipe is positioned in front like this, and there is a flap here, and this one is called the epiglottis. It's called the epiglottis. This flap regulates whether a particular substance is going to enter into the trachea or enter into the esophagus. So when you are swallowing, this epiglottis is going to cover the entrance into the trachea, because it's a trachea, or we call it the windpipe. Okay, and at the back here, you have the esophagus. So the food is going to push down into the esophagus here. It comes down. And as the food comes down, the walls of the esophagus are a bit muscular, okay? Because it contracts and relaxes. There's a rhythmic contraction and relaxation that takes place. And the wall has 4 layers. One, we have the mucosal layer. 2, we have the submucosal layer. 3, you have the muscular layer. We call it the muscularis. And then the 4th one, we have the adventitia. So these are the 4 layers of the esophagus. You have the mucosal, the submucosal, the muscularis, and the adventitia. Now, let's say here, a food is being pushed down, because this is how the bolus move. This is the bolus, the food that the tongue has rolled. So as it is coming down, what happens? When it leaves this point and comes to this point, this end here is going to contract. This end will contract, propelling the food downward. and this end will open up. So as the food enters here, what happens next? This end also is going to contract, pushing the food down. So this contraction and relaxation of the walls of the esophagus, we refer to it as peristalsis. We call it peristalsis. So meaning the food moved by a means of a peristaltic movement, which is as a result of the rhythmic contraction and relaxation. The rhythmic contraction and relaxation of the walls of the esophagus. It pushes the food downward into the stomach. As the food is pushed down on its way into the stomach, there is a separation between the esophagus and the stomach. Now, here, this is the bolus coming down. The bolus is coming down. So as the bolus approach this point, there are two valves here. And these valves, we refer to them as the cardiac sphincter. These ones here separate the esophagus from the stomach, because looking at the stomach, it is a pear-shaped muscular organ. This is the stomach. A pear-shaped muscular organ that is also made up of 5 layers. And what are these 5 layers? The first layer, we have the mucosal, you have the submucosal, you have the muscularis, you have the subserosa, and then finally you have the serosa. These are the 5 layers of the stomach. This is the innermost. This is the innermost layer. And this here is the outermost layer. Meaning this is from inside to the outer part. So 5 layers here, which are going to help in the churning of the food. And inside the stomach, here, you have very important substances that are produced. One of the substances you have is gastric juice, which consists of 2 enzymes. One of the enzymes is pepsin, and the other enzyme is renin. So you have pepsin and renin that are produced, and the other substance produced inside the stomach is hydrochloric acid. Which is HCl. Now, this substance here is going to neutralize the food coming from the mouth, because the food from the mouth is alkaline. The particular enzyme we discussed in the mouth, which was amylase, can operate in an alkaline medium. Okay, so meaning the juice that is produced inside the mouth, which is saliva, is alkaline in nature. So it is going to be neutralized by the acidic content of the stomach. And one of the functions of these substances, which is the HCl, one is to kill or make any bacterial inactive that is entering with the food into the stomach. It will either kill the bacteria or it makes the bacteria inactive, okay? And then it prevents the food from decay, and also it helps in the neutralization of the alkaline substance entering the stomach. This HCl, which is the hydrochloric acid, is secreted by oxyntic cells. This one is secreted by oxyntic cells of the gastric glands. So the cells of the gastric glands, which secretes the gastric juice here, containing pepsin and renin, and then also hydrochloric acid. Pepsin is secreted as pepsinogen. So it is the hydrochloric acid, the HCl, that converts it into pepsin, which is an active substance. Renin also is secreted as prorenin. It is also the HCl that converts it into an active renin. So these ones here are inactive. This is inactive. This one also inactive. So they are activated by the HCl. So the HCl activates it into pepsin, and this HCl activates the prorenin into renin. Okay. Now, what is the role of pepsin here? Pepsin is going to help to hydrolyze proteins into short chains, polypeptides. Okay, so they are going to be hydrolyzed into polypeptides. So pepsin here is going to hydrolyze proteins into short chains polypeptides, whereas renin helps in the coagulation, meaning it's going to solidify. It is going to coagulate. In the coagulation of soluble milk proteins we call carcinogen. So these soluble carcinogen here are going to be solidified into caseins, and which are later acted upon by pepsin. So pepsin further hydrolyzes them into short chains polypeptides. So meaning the enzymes inside the stomach act purely on proteins. Pepsin hydrolyzing proteins into short chain polypeptides, and then you have renin for the coagulation of the soluble carcinogen into casein, which is a solid substance. So this is soluble in casein here is insoluble. Okay, we call it solid. So the presence of food inside the stomach is going to stimulate these gastric glands into producing more gastric juice. Okay, and the production of saliva in the mouth is also stimulated by the mere sight of food, or by the smell of food, or just by the expectation of food. That's what happens there. So inside the stomach, when this has taken place, the stomach is going to continue to contract and relax. And as it contracts and relax, it mixes the food into a semi-solid. And that semi-solid, which the food is mixed into, we refer to it as a chyme. So this chyme is a semi-solid. So meaning we no longer refer to it as maybe starch or proteins or whatsoever. We now refer to it as a chyme, a semi-solid, which has been blended up. So this chyme, a semi-solid, is going to be pushed down further into the alimentary canal. There are ring-like structures which are muscular here, and these ones we refer to them as the pyloric sphincter. So from this point, the food enters into the main seat of digestion in the human alimentary canal. Now, inside the duodenum, you have the liver. And the liver, of course, here you have a leaf-like structure called the pancreas, and which is connected into the liver being the heaviest internal organ. Now, this liver here produces a bile, and this bile is produced from worn out or dead red blood cells, the RBCs, meaning dead erythrocytes. So it is from those that the bile is produced. And this bile here is stored inside the gallbladder. This structure here is the gallbladder. That is where the bile is stored. And this bile consists of salts and a pigment. One of the salts it has is sodium taurocholates, and glycocholates. So these are the salts which the bile contains. And it also contains pigment, that is bilirubin and biliverdin. That is bilirubin and biliverdin. These are the pigments. And the function of this bile is to emulsify fats. So it helps in the emulsification of fats. It emulsifies the fats. To emulsify means it is going to break down the fats into tiny droplets, like how you have detergent acting on fats. How detergent acts on fat, that's the manner in which this bile acts on fat. Mind you, the bile does not contain any enzyme. It has no enzyme. It only has bile salt in the bile pigment, and the salts are sodium taurocholates, and then you have glycocholates. They are the salts, and you have the bile pigment, which is the bilirubin and the biliverdin. These are the pigments that are found inside the bile. Now, from this point, mind you, the food leaving the stomach, entering into the duodenum is acidic. That is the chyme. The chyme entering there is acidic. The bile also contains sodium hydrogen carbonate, so we call it sodium bicarbonates. The bile contains another salt, which is sodium bicarbonate, that is NaHCO3. This is the salt which it also secretes. And this guy here is, we know it's an alkaline substance. It is alkaline in nature. Okay, so it is going to neutralize the acid. It neutralizes the acidic chyme. The acidic chyme, because we no longer call it food, we call it a chyme. The chyme leaving the stomach and entering into the duodenum, the small intestine. So it is going to neutralize it from the acidic substance into a neutral substance. Now we talk of the pancreas. What happens there? This pancreas here produces a juice we call the pancreatic juice. And this pancreatic juice consists of three enzymes. So this pancreas opens into the pancreatic duct here and further opens into the bile duct, which opens into the small intestine, this coiled structure here. Now, it opens into it. This pancreas containing three enzymes. One, we have the pancreatic amylase. The pancreatic amylase is also known as diastase. And then you have trypsin, which is secreted as trypsinogen. Diastase here, or we call it the pancreatic amylase, is going to act on starch. It hydrolyzes the starch into maltose. Okay, it hydrolyzes into maltose, which is further going to be acted upon by maltase. And maltase is going to convert this maltose into glucose. Yeah. Maltase converts the maltose into glucose, which is the end product of the digestion of carbohydrates. Okay. And then we look at the trypsin, which is an endopeptidase. Trypsin here is secreted as an inactive substance. We call it an inactive trypsinogen. So this trypsinogen is going to be activated, converted into trypsin by the enzyme enterokinase. Enterokinase here is going to activate the trypsinogen into an active trypsin. And this active trypsin here is an endopeptidase. It is then going to convert proteins. It hydrolyzes the proteins into polypeptides. So the proteins are hydrolyzed into polypeptides. Okay, so this is the hydrolysis that takes place. And we look at the pancreatic lipase. The pancreatic lipase is going to act on fats and oils. It is going to convert them into fatty acids and glycerols, which is the end product of lipids. So from that point, the lipids can be absorbed. So pancreatic lipase, these ones are going to hydrolyze. So they are responsible for the hydrolysis of fats and oils. So they convert these fats and oils into fatty acids and glycerols. So they convert them into fatty acids and glycerol. And this is the end product of the digestion of lipid. So from this point, they can now be absorbed into the bloodstream. So absorption can easily take place here. So next, we look at the secretory cells. These secretory cells are going to produce mucus. They produce a digestive juice, which consists of variety of enzymes. And one of those enzymes is maltase. Maltase is going to convert maltose. Here, it is going to convert it into glucose and fructose. So it converts the maltose into glucose and fructose. We also have lactase. Lactase is going to convert lactose, and lactose is converted into glucose and galactose. And these are the end products of the digestion of carbohydrates. So maltase converting the maltose into glucose and fructose, and then lactase converting lactose into glucose and galactose. Okay. In the third one, you have epipsin. Epipsin here is going to convert the polypeptides. It converts the polypeptides into amino acids. Okay, so this is what happens inside the duodenum. So we should know that the secretion of most of these things are controlled by the nervous system, and it's as a result of nervous reflexes that brings about the secretion of these enzymes that we've looked at. So now we know that the small intestine is divided into three parts. You have the duodenum, you have the jejunum, and the ileum. Now we've looked at what happens inside the duodenum. Now inside this jejunum, no digestion takes place. So after the conversion, after the mixing up of the chyme from the stomach into the small intestine, after the blend, you are going to have a waterish emulsion. And that waterish emulsion, we refer to it as the chyle. So meaning food in the small intestine is called chyle. Mind you, inside the stomach, we refer to it as the chyme. And now here, we call it the chyle because it is now waterish. So it is going to leave the duodenum and enter the jejunum by means of peristalsis. But here, no digestion takes place, and then it comes into the ileum. The ileum is coiled. So we're going to look at the walls of the ileum. What happens to the food inside the ileum there? Now, in the ileum, it is coiled and it has a large surface area. The walls of the ileum are about 3.6 meters long, and they are coiled. It's very long, so that longness gives it a large surface area. And it is in the ileum that absorption, a very important process, absorption of food substances into the bloodstream takes place. And this is possible as a result of its length and large surface area. And it has projections there. Those projections, we refer to them as the villi. Now we're going to look at the structure of a villus, because this is the plural. So one is villus. Now this is how the structure looks like, okay? Now this outer part here is the epithelium. Which is made up of epithelial cells. And in here, you have the lacteal. And these are blood vessels. These are blood vessels here. Because the food, the digested food substances now, the glucose, the fructose, the galactose, the amino acids, the fatty acids, and glycerols are going to be absorbed here. These are the digested food substances. Okay, this is how they enter, because this one is also made up of four layers. You have the mucosal, you have the submucosal, you have the moxularis, you have the muscularis rather, and you have the serosa. So those are the four layers of the lining of the small intestine. So these ones here, this is the lacteal, and then these are blood vessels. These ones are blood vessels. So this is the epithelium, which we refer to as the epithelial cells, which are having microvilli cells. And this is the lacteal. These are blood capillaries. They are blood capillaries. Okay. And then you have this portion here, this mid piece, that is the lacteal. And this here tells us it is the continuation of the blood capillaries. And here you have nutrient rich blood. There's nutrient rich blood. So blood that is very rich in nutrients, and this tells us of the blood flow. Okay, this is the blood flow. So this here is how the food substances, the glucose, fructose, the galactose, the amino acids, the fatty acids and glycerols are going to be absorbed and assimilated into the bloodstream. So you have millions of these which are found inside the small intestine, because we said the small intestine is long with a large surface area, which you have a lot of villi from there. So this is just one villus magnified for you to see what happens inside there. You have also these capillaries, which are responsible to carry deoxygenated blood into the venules, leading into the hepatic portal. The means through which food substances can penetrate into this is by means of diffusion and that of active transport. And when the nutrients are being absorbed into the bloodstream, now a leftover of the unabsorbed food is going to move down, and it moves down into where the colon. The colon is known as the large intestine. So here, this is just a look at the digestive tract. Okay, there's the human alimentary canal, how it looks like. So now this undigested food, the one that didn't absorbed, the one that didn't absorbed into the small intestine is now going to be sent here. It is sent into a structure we call the caseum. Okay, and the caseum here is not that active in man. It is active in other ruminants, like in cattle and all. And then the caseum extends into what we call an appendix. And this appendix is a vestigial organ. When we talk of vestigial organs, these are organs that are of no relevance in us. They are of no importance at all. So this colon here, this is the first portion, which is the caseum. Okay. And this caseum, when the food has come here, it is now going to ascend. So this first portion of the colon is called the ascending colon. This is the ascending colon, because to ascend means it is moving up. And this is where more water is going to be sucked from the chyle. So the absorption of water takes place inside the large intestine, that coil tubule. So from there, it is pushed into this other end of the colon. This is called the transverse. That is the transverse colon. And from there, it is sent downward into the descending colon. And there is a smaller branching hair comes that is called the pelvic colon. So through this hair, it is water that is going to be absorbed. So I call it negative water. More water is going to be absorbed and solidifying it into a paste. Okay. And that paste, which we refer to as the phases, and then it is going to be stored in here, and that is the rectum. And inside the rectum, you have gut valves. Those valves, we call them the intrinsic and the extrinsic valve. Intrinsic. This one is the internal one, and you have the extrinsic. This one is out, the external one. So these are the ones that prevent us from embarrassing ourselves in public, meaning as soon as the feces or the undigested food are sent into the rectum, it does not just come out of the anus, because this is the anus. This here is the anus. So it does not just come out. It is controlled in that place, okay? And then it is going to be there for some time. And then when you're ready to give it out, we call that process defecation. You defecate. Okay? So you defecate. So defecation takes place. And you defecate phases. And also, there is a sickness that is associated to this end. Now this appendix here can get ruptured. And what causes the appendix to get ruptured? Now, when the chyle, which is leaving the small intestine, enters into the caseum, it is going to enter alongside some heavier particles. Particles like the seeds of pepper, stones that may have entered accidentally with the food inside the small intestine, and other heavier particles are going to remain here. So during the ascension process, as the food is ascending and more water is being sucked from it by the colon here, those heavier particles are going to remain inside the caseum, and they are going to be deposited inside this appendix. And when this collection becomes too large, it causes the appendix to perforate. Okay, the appendix can perforate, and then that sickness we refer to it as appendicitis. And when it gets ruptured, it can be removed by a surgery. Okay, it can be removed by a medical practitioner. This is it for the digestion in man. Thank you very much for watching our video. Subscribe to our channel if you are new. Like our video. Hit the notification bell to be notified each time we release a new video. You can like our page and follow us on Facebook, Jalo Science Academy. There's also a study group program, which is a channel, Jalo Science Academy. You can join the channel and be part of us, where you can direct your questions and we will gladly answer them for you. Of course, share this video to your friends so that they can benefit. And don't forget, never stop learning, because those that have stopped learning have stopped living. Thank you very much.